happy to be here. So today I would like to present a tech talk on the pan-cancer assay-ready organoid drug screening platform of Crown Bioscience. So hi, my name is Liza. I work as a scientist in the in vitro RNA department at Crown Bioscience, the Netherlands. So welcome to this tech talk. Today I'll be giving you some more insight on our large organoid panel drug screening. And so let's get started. So the aim for Crown Bioscience with launching this new technology is to move oncology models closer to the clinic. So there is an increased need for patient-derived translational models because despite advances in both 3D in vitro human cell-based models and also in vivo animal models, currently 97% of oncology drugs that enter clinical trials fail to end up being approved for use in the clinic. So to overcome this very poor approval rate, we really need patient-relevant translational systems that mimic the heterogeneity and molecular and also genetic complexity of these tumors. So we can make better informed decisions about progressing which drugs we need to progress into the clinic, to test drugs on the most relevant and also predictive models, and also to understand the drug effects on the native tumor microenvironment. And also at the services of Crown Bioscience, we can evaluate immuno-oncology drugs, including immune checkpoint inhibitors. So to that end, we use our organoids for in vitro patient avatars for drug responses. So over the last couple of years, there has been emerging literature and evidence that indicates that organoids can be used to accurately predict drug responses. So they are phenotypically and genotypically profiling of original patient tumors. And also the molecular profiling suggests that tumor organoids can complement existing approaches to define cancer vulnerabilities and improve treatment responses. So to that end, we can exploit organoid technology for translational oncology. So these tumor and also their paired normal organoids have very stable genomic and epigenetic fingerprints, and including clinically relevant mutations. They make the ideal model for drug screening, efficacy testing, and also genetic engineering studies. So what we know from organoids is that they are highly clinically predictive. The only in vitro platform using clinically relevant models for high throughput screening, and they are very much predictive of the patient response. They are also easily scalable. We have generated large biobanks of these organoid models. We can test multiple drugs and combination of drugs in one assay, and we can profile multiple models to capture the patient diversity. And also we have the ability to genetically engineer the models that are still lacking from the patient population. Moreover, they have superior efficiency. There is a very high signal-to-noise ratio compared to standard 3D models, such as spheroids. And also they have a very low batch-to-batch variation. And they are highly reproducible compared to primary models. And then in our Crown Bioscience platforms, we also have matched in vivo PDX models for the majority of our organoid models. Moreover, we offer biomarker evaluation, and we can bridge the transition from the in vitro testing to in vivo testing. So I already briefly touched upon it. So we have a large set of matched pairs of normal and tumor organoids. So here you can see an example of matched colorectal tumor organoids and their normal counterparts. And also they capture a large variety of signature mutations in colorectal cancer. So within our living biobank of these organoid models, We have now included at least 441 PDX-derived models and 215 patient-derived organoids, or PDOs. And they reflect the genomic, morphological, and physiological characteristics of their corresponding organ and tumor in vivo. So here you can see the different indications that we currently have in the biobank. However, the biobank is constantly evolving, and more models are being added. And for very rare types of tumors, we have the possibility also genetically engineer models that are still lacking from the biobank. So at Crown Bioscience, we're advancing in the in vitro drug screening platform by using automated processes to really improve efficiency and also the turnaround time. We include multiple negative and positive controls to control any inter and intraplate variation. We have our standard cell-death glow readout, and we also look at morphology. And of course, additional readouts are always available on request, such as apoptotic assay cell cycle investigation and also biomarker analysis. So we have a standardized analysis and report format, which we can use to efficiently bring the data as soon as possible to our clients. So on the right, you can see multiple liquid handlers and equipment that we're using in the large organoid panel screen. So here you can see it a little bit more in depth. So prior to creating the panel, we perform organoid expansion. So we create the batches of assay-ready organoids. And then on the day that we actually start the assay, we seed all the different models at the same time. We're able to efficiently do compound addition with our liquid handlers. And then after a fixed amount of time, usually after 6 days, we perform the cell-death glow readout. And then the data is analyzed in a very efficient manner, and we can deliver the report to the customer in a time span of only 6 to 8 weeks. So after our standard readout of cell-death glow cell viability analysis, we also offer a large service of more in-depth analysis, such as the high-content imaging-based high-throughput drug screening platform. So we are highly specialized in these specific techniques. So libraries of those ranges of small molecules, combination therapies, immuno-oncology therapeutics, bispecific antibodies, drug conjugates, and inflammatory agents, and also toxic agents can be screened in the high-content imaging platform. So this platform generates 3D imaging, and we can analyze it in our OMiner software, where we can generate up to 500 different parameters for not only assessing the drug response, but also seeing co-localization, for instance, of the antibody, but also observe small changes in morphology, and also to characterize these treatment effects on organoids with specific mutations and diverse genetic backgrounds. So again, we have a lot of models that are suitable to use in our high-content imaging-based drug screening. So to assess the quality of our assays, we have our organoid platform performance data presented here. So on the left, you can see the Z factor, in the middle, the control variation, and on the right, the assay window that we assess for each model that we test in the high-throughput screening setup. So according to industry standards, the Z factors above 0.3 are considered acceptable, and above 0.5 they are considered to be of high performance. As you can see on the left panel, 90% of the models that we've tested so far in our organoid platform are actually of really good quality. And also the control variation, as shown in the middle panel, the industry standard is below 15%. And as you can see also here, the majority of the models fall within this range. As you can see, there is one model that is definitely not meeting our quality standards. So with this assessment. After we perform the assay, we are then, of course, excluding this data and repeating the assay. And on the right, you can see the assay window. So this is a measure where we can actually see how well the organoids are proliferating in the assay. So the higher the number, the bigger the proliferation rate. And also here, the industry standard is above a 50-fold of assay window is considered a good assay window for assessing the drug efficacy. And go. So here you can see the reproducibility of the data over different passages and also using different technologies. So in the A panel, you can see the drug response of a classically prepared and also assay-ready organoid. And then you can see the drug response to standard of care SN38. And as you can see, all the graphs are matching each other. So there's no difference in drug response between the classic and the assay-ready platform. Then in panel B, we again see the drug response to SN38 for the same organoid model, but then over different passages. So passage 19, 20, 21, and 29. And we can actually see that the expansion and long-term expansion of this model does not affect the drug response. And then in panel C, we can see the drug response to 5FU when we use different liquid handlers at different locations of Crown Bioscience. And here you can actually see that with the high performance of our liquid handlers, there's also no difference between the different locations. And then also, if we look at the IC50 for the SN38, we can actually see that the IC50s are very comparable between the different passages. So in our platform, we offer different readouts. Already briefly mentioned the high content imaging and our standard cells at the glow readout. And here you can see that with a different technique, we are actually able to assess the drug sensitivity of the same model with a different technique, which results in similar outputs. So on the left, we have the high content imaging. It produces over 500 different parameters, but if we select the correct ones to assess the cell viability, we get a very similar output to the cells at the glow readout. And then the high content imaging platform provides a lot of additional information next to just relative cell viability. So we can actually visualize the number, size, and shape of nuclei, the cells and tumoroids. We can visualize specific subpopulations. We can assess apoptosis, invasion, and also in combination assays, we can assess immune cell infiltration and tumor killing. And also we can really dive more into the phenotype of interest. With the high content imaging platform, we can assess therapeutic responses and also investigate the mode of action and off-target effects. So on the right, you can see some of the images and also the automated analysis tool that we use. And then on the right, you can see the drug response curves that are generated with the high content imaging data. So by using our preclinical pipelines, we can actually fast track the full drug discovery pipeline from the lead compound to the mechanism of action. So here a paper is shown from Herpers et al. And they actually screened for a specific for a bispecific antibody, and they used organoid technology for the full in vitro preclinical pipeline. And then actually this in this particular case, the mechanism of action in vivo could be skipped and they could directly go into the clinical trial phase, which is a major advantage for shortening the timelines from selecting the lead compound to actually getting the compounds approved and having it being efficiently used in the clinic. So then if we perform our large panel organoid screen, this is an example of how the data can be represented. So here you can see the IC50 values of paclitaxel, gemcitabine, and cisplatin for 50 models of the panel. And here you can see that there is a wide response, a wide difference in response from sensitive to more resistant models. Then also in our large panel organoid screening, we aim to have a full scope of different mutational profiles that are clinically relevant, such as the KRAS mutation. So here you can actually see that upon testing the targeted therapy AMG 510, which is aiming at the KRAS G12C mutants, then we can actually see that those models are very nicely clustering together, whereas the other KRAS mutations and the wild type are not showing much response to the KRAS AMG 510 drug. So again, if we look at the area under the curve here, we can see that the G12C models are clustering together, whereas the other non-specific or non-targeted mutations are nicely clustering together with the wild types. So then, talking about the application of our large panel organoid screening, we can offer a preclinical in vitro clinical trial where we can actually screen a large set of indications. So for further research and further testing, we can select the right indication where the drug is effective, and we can also really support the drug repurposing movement that is currently ongoing. And also we offer the biomarker in identification and also the selection of models for further in vivo follow-up. So just to conclude with an overview of our different models, so we have the 3D tumoroids, we have a large biobank of these PDX-derived organoid models that contain over more than 23 cancer types, and also have the matched in vivo PDX models, together with our hub patient-derived organoids, which are also featuring normal and tumor matched pairs, and then our 3D ex vivo patient tissues, where we can directly test fresh patient tumor samples directly into the assay plate and perform the assay with preserving the native tumor microenvironment with all the endogenous immune cells, fibroblasts, and stroma. And with that, I would like to end my tech talk. Please reach out to us if you have any questions. Reach out to sales@brownbio.com, or feel free to contact me for further questions.